it is more than 80% of all land plants form this symbiosis. And as the name suggests, the Greek myco and rhizo literally means fungus root. There's different types of mycorrhizal fungi, and I put here endo and ecto. These are broad categories, which again, as the name suggests, refer to the place where the interface between the fungus and the plant occurs. So endo, the interface for exchange is occurring inside the root, and ecto, it's outside the root cells. Or inside the root cell, so cortical cell, and outside the cell. And they're important because it's a win-win for both organisms. So the fungi, they gain an energy source in the form of sugar, carbohydrates from the plant, and in exchange, they give the plant increased access to water and nutrients, and can also provide protection against root pathogens, environmental stress, and so forth. So the earliest fossils of land plants include structures in the roots that appear to be mycorrhizas. So it's now thought that the evolution of plants on land occurred because of this symbiosis. So the fungi allowed plants to colonize land in the first place. It began with what is still the largest group in terms of the plant species richness that it associates with. That's the arbuscular mycorrhizal fungi. These are fungi that occur within the cortical cell of the plant root, and today are most important in agricultural systems. So a lot of annual plants, annuals, some forbs, and also some trees and things, but especially the grasses, the annuals, they associate with arbuscular mycorrhizal fungi. And it's important to point out that this is a completely different animal than some of the other types. So they're classified according to this interface structure between the fungus and plant where the exchange takes place. But at higher taxonomic levels, they're quite distinct organisms. Also, they evolved along with the plants then into more complex structures. Some of these shown here might have originally been thought to be mycorrhizas because they mimic mycorrhizal structures, but then were later found that actually they're not benefiting the plant. It's just maybe rudimental structures that appear to be mycorrhizas. So there's been some reclassification over time. But in general, you have these broad categories of arbuscular mycorrhizal fungi, and then you have other types that grow inside the roots of orchids, of ericaceous plants, and finally getting into the ectomycorrhizal fungi, which the interface is occurring between the epidermal cells, so between and sometimes into the cortical cells of the plant root. And some plants have also evolved away from the symbiosis. So although the earliest fossils, they all appear to have the symbiosis, plants have also evolved other nutrient acquisition strategies. So some plants are not mycorrhizal, but this is usually the exception, and it usually occurred later in evolutionary history. Vice versa, fungi have also evolved independently into the mycorrhizal habit. So different lineages of fungi have come to become mycorrhizal independently in convergent evolution. This figure just shows the major sequences of events, so the main radiations of diversity within these types, showing more than 50 million years ago with the establishment of plants on land. And then there was a second large radiation with the occurrence of the gymnosperms about 150 million years ago. And then more recently with the rapid radiation and diversification of plants, this is also mirrored with further diversity among the mycorrhizal forming fungi. This shows it again by host plant type. So this is a bit biased, this pie chart, by species richness. So you see the vast majority of plants form arbuscular mycorrhizal fungi because that's also the most plants on the planet. Orchid mycorrhizae, ericoid mycorrhizae, they form lesser percent. Plants which have evolved strategies here, they're grouped into habitat specialists and nutrient acquisition specialists. So like predatory plants, for example, plants that occur in bogs where there's low oxygen levels. These habitats still hold mycorrhizal associations too, but they tend to have more plants that don't have mycorrhizal associations. If you're in the agriculture sector, so the Brassicaceae is a typical example of a plant that no longer has mycorrhizal plants. You see these huge raps fields, and in these fields there's no known mycorrhizal associations occurring, for example. As I mentioned, there's a cost benefit. It's a win-win for the symbiosis, but it comes at a cost for both partners. So you get a whole spectrum from mutualistic to parasitism. Can depend on the life stage of the plant, the environmental conditions, and so forth. But in general, it occurs and is evolutionary stable because it's a win-win for both partners. It's worth the investment. The vast majority of the mycorrhizal symbioses occur here. They're facultative mutualists, so they're about 50% of the ectomycorrhizas, for example, are generalists. They can associate with multiple different plant hosts. Whereas getting into obligate mutualists, it requires more investment and you have fewer of them, but they do occur. There's also plants that, at least in some parts of their life, have learned to cheat the system and take advantage of, say, the fungi. Microheterotrophs are an example of this, and they're listed here down on this axis with the exploitative side. So they typically trick the fungus into colonizing it. They're getting sugars, nutrients, other things from the fungus in exchange for nothing. So they're not giving anything back, at least for the majority of its life. That fungus is still getting carbon, though, from other plants. So example, microheterotrophs are like the monotrophs, a lot of terrestrial orchids, any achlorophyllous plant. A lot of these originally were thought to be epiparasites growing on other plant roots. As it turns out, most of these have very specific relationships with the mycorrhizal fungus. That fungus is in turn a generalist that's maybe connected to a pine tree or some other plant in the overstory. And it's making a network where the achlorophyllous plant is not only getting nutrients and water, but it's also getting its carbon and sugar through this fungal pathway, through the photosynthesis of the overstory plants. And then finally, some plant hosts can form multiple different types of mycorrhizal associations, both arbuscular and ectomycorrhizal. And it's not really known why, if it's just opportunistic and they get colonized by anything they can, or if there's some evolutionary advantage to this as well. But what the studies that have investigated are finding is that this really depends on environment, and it can also depend on the life stage of the plant in terms of the proportion of which type of mycorrhizae it has, or is the most common. Examples are acacia, eucalyptus, even oak and maple. I don't have the species name or the Latin names listed for those of you that are German speakers. Maybe you know some of these common names also in English, but a significant portion of trees actually do, of deciduous trees especially, have both. And this is figures just to show that it's complex. So it's not straightforward. That is, it's never 50-50. It can depend on the context, which mycorrhizal system wins in that particular environment. And what I'll be mostly focusing on today is the ectomycorrhizal group. So although these only represent 2% of the associations with plants on Earth, more than 60% of trees form ectomycorrhizal relationships. So of course they have an outsized impact on global ecosystems, especially temperate forest ecosystems. Getting into tropical forests, you have more mixed types, more arbuscular mycorrhizal associating trees. But in temperate forests, almost all the trees are associating with ectomycorrhizal fungi. Cedar is one exception. It forms arbuscular mycorrhizal associations. So, an ectomycorrhizal fungus, again, these types are usually characterized by the interface of the plant and the root, and that is shown here. The root and the fungus, that's shown here. And as most of you probably know with the macrofungi, so a lot of these fungi that are forming ectomycorrhizal associations also form mushrooms in their sexual stage. And most of you probably know that a mushroom is like the apple on a tree. The majority of the organism is radiating below ground in filamentous hyphae, collectively referred to as a mycelium. And these hyphae then enter the root. So the symbiosis is initiated when they meet. They're sending out chemical signals. They recognize each other as a compatible partner and start to grow towards each other with chemotaxis. So one of the controversial things you'll see maybe in the popular media is plant communication. Do plants communicate? How do you define intelligence in plants and things? Well, they're certainly communicating. You know that because you can watch it happening. You can watch them growing together. They're sending chemical signals, and they're recognizing each other's signals and responding to that. And that, in a sense, is communication. So once it's formed, the hyphae intertwine around the epidermal and cortical cells of the root and form what is called a Hardig net, or a mantle around the net. And it significantly changes the behavior and the appearance of the root. They swell up, they can get funky shapes and different colors, be shiny, all kinds of different shapes, colors, forms, and functions. But my point is that it significantly alters. It becomes a different organ. It's no longer a root, it's no longer a fungus, it's a symbiotic, it's a different structure. Then outside, away from the root, up here, if you look in the upper left for you, then you have the extramatrical hyphae, what's radiating out. You have hydrophilic hyphae that is going out and exploring the soil, and the hyphal front, as they call it, that's what's exuding enzymes, excreting enzymes and collecting nutrients and water and things from the soil. Some ectomycorrhizal fungi can also form very highly specialized tube-like channels out of these hyphae called rhizomorphs. And the rhizomorphs themselves can have very different structures depending on the species. But these are hydrophobic and are particularly adept at transferring water and nutrients over significantly meaningful distances. So you have these different parts of the fungus that's out exploring the soil, moving things around in the soil, and associating with the plants that it encounters. And usually once they join and grow together, then they also coordinate their growth some as they explore the soil then together, the root and the fungi. I'll just point out here, you see a picture of a chanterelle. So a lot of very important edible mushrooms are also ectomycorrhizal. So the Steinpilz, the porcini, Pfifferlinge, or chanterelle shown here. In other countries, well, truffles also here, truffles, false truffles. The matsutake mushroom, very important economic, is also ectomycorrhizal. And not only for us, but also for small mammals. Love to eat mushrooms. Small mammals, large mammals, wild boars, deer, bear, many mammals that you don't even think of can actually have a surprising amount of fungi in their diet. And vice versa, it goes on up the trophic scale. There's birds that depend on those small mammals and so forth. So very important for the ecosystems in different ways. I mentioned that they have a lot of different forms and functions. And these can also be used as a classification system. Not only how the root tip looks, the ectomycorrhizal root tip, but also how they explore the soil. How much extramatrical free radical hyphae they have. So how much, some are very localized, some are small, some are large, some have a lot of rhizomorphs or highly specialized rhizomorphs. others not, and so forth. And these can be used to put them into general categories, which then is basically a proxy for possible functional traits. Oh, and I'll point out the researcher that spearheaded this, more or less, was also a German researcher in Munich, Reinhard Agerer. He's now retired, but he did a lot of the morphological classifications of ectomycorrhizal fungi, and also proposed this system of classification based on the exploration strategies, which is quite useful for, yeah, you can get a lot of information without a lot of money or time needed for the more detailed analysis to get at the identification of the fungi. So this is just to remind you that how diverse, and I think they're quite spectacular. I mean, if you think about all the different shapes and colors of the the sporocarps, what we see above ground, the mushrooms, this is basically mirrored below ground. And I have to admit, not all these mushrooms are mycorrhizal. I slipped a few in there just because they're colorful and glowing and things, but you get the picture. It's diverse. And it's the same below ground. A single tree can have 100 different types of these fungi on it, and typically does have 100 different types unless it's growing in a city, but that's a story for another time. I like to compare it to a coral reef. One difference is that we know much more about the world's deepest oceans than we do 20 centimeters below our ground in terms of what's really going on there and the true diversity and the spatial distributions and everything. But we know that it's diverse and it's quite spectacular. I think I liken it to a coral reef, but you have to think about that's sort of looking at it like just at the root interface. And then you have everything going on in the water, and that would be the soil. So now you have this complex soil matrix where all this biomass of, that's where the mycelium and the hyphae is running around and all the other soil organisms and just the complex, biological soup. It's not even a biofilm because it's like, so three-dimensional and so much biomass and stuff. It's really just it's constantly moving around. There's turnover, there's competition, there's some explore more, some stay put. There's a lot going on. Very dynamic system. And because I assumed that you would be more familiar with the trees, because most people are, that's what we see above ground. That's how ecosystems or forest ecosystems are often defined is by the trees. It's the foundational species. It's what we see, and they're long-lived. So I wanted to say a few interesting things about the fungi from the biological perspective. And these might be things also that are important to note if you ever experiment with ectomycorrhizal fungal systems, because they can create problems. Even things like, what is the concept of a species? So with ectomycorrhizae, I'll talk more about that in a moment, but if you look at some of the other mycorrhizal types like arbuscular, then it's sometimes even more vague because they don't have a sexual reproductive stage, but they're still very diverse. And it seems like things are just, how they're recombining and diverse, but yet still maintaining, identifiable species and stuff over time is also a lot of unknowns. But with the ectomycorrhizal fungi, these are mostly formed by dikaryones, and a dikaryotic organism is sometimes for most of the life cycle, sometimes just shortly, but it's basically you have two haploid nuclei existing in the same cell. So two distinct genetic, signatures, haploid nuclei in the same phytoplasm, but they don't fuse, so they don't join into the nucleus. And it's basically, if you, from basic biology, you have plasmogamy without karyogamy. And why is that important? Well, they can actually, it's been shown or found that in some environmental triggers or certain environments, they can actually, outside of the sexual reproductive stage, they can recombine DNA between these two distinct haploid nuclei. And in this way, they can evolve very rapidly to adapt to changing conditions or to stress. It also leads to genetic and phenotypic plasticity. So phenotypic plasticity is especially important when we start to talk about forest ecosystem and the relevance for forests. Yeah, but it goes beyond this too, because not only do you have dikaryosis, heterokaryosis, but some of these fungi, you know, the hyphae are exploring the soil. They encounter each other, they fuse, you know, or they don't. Some can actually fuse with other individuals and form more or less a superorganism. And so now you not only have two haploid nuclei in the same cell, but you can have, you know, this part of the organism has A and B, and this part has B and C, and this part has C and A, and so forth. So you have this like compound mycelium, which is actually comprised of different individuals. Matsutake was the first study I know of that really showed this in the mycorrhizal fungi. It was really cool. They found that you know, Matsutake grow in fairy rings, and they get bigger and bigger and bigger in the hyphal front over time. And as they get bigger, they encounter other individuals, but they don't compete and necessarily wipe them out with the same species I'm talking. They can simply fuse. Anastomosis, they join their hyphae together, become a single organism, and then they coordinate their growth. They continue growing as a fairy ring, where the center is free, so there's no mycelium in the center, but it's sort of in a ring, and they continue growing out and out like this. And just last year, this was also shown for truffles. It was in France or Italy. A study found that truffles were also forming these compound mycelium, where you know, you might have a tree and they're collecting the truffles for the market, but they're actually different, unique individuals, all fused together into a super organism, more or less. And that's chimerism. You might know it as chimerism, where you maybe have your hand has a different genetic fingerprint than your head. Some trees do this and stuff, but it's usually more limited. Fungi are quite good at this. They're heterotrophic, so they absorb nutrients and what they need from outside their bodies. They release enzymes and then suck them back up through the hyphal straws, more or less. Another unique aspect is their cell walls are made of chitin. And this is a defining characteristic actually in the kingdom fungi. I think so. I don't think there are any exceptions, but yeah, so similar to insects, arthropods, they have cell walls, mostly made of chitin, which is of course very recalcitrant, even more so than cellulose cell walls like in plants. So very, yeah, robust critters. The spore, outside wall of a spore, is the hardest known organic compound known in nature, to occur naturally in nature. And these can also withstand extreme pressure. They can survive extreme radiation. They can travel in outer space. And yeah, and therefore the space station has a mold problem. You know, there's not a lot of other organisms that become invasive species on the International Space Station because the conditions are so rough. But mold does pretty well. And we need to pay attention to this because that means that when in our space travels, you know, we have hitchhikers that are able to survive these environments, and we could be contaminating these foreign environments through our space travels. So now they are more careful, the you know, the space agencies and stuff, than they used to be. The late Terence McKenna, I don't know if some of you, if you're familiar with the ethnobotanist Terence McKenna, he actually proposed that they originated from outer space. To my knowledge, there's no direct evidence of this, but theoretically it's possible. And some are still so strange to science genetically and how they behave and things, and just so mysterious that it wouldn't surprise me, to be honest. So enough about the biology, though. How are we doing on time already? I want to also talk about some points that I think are generally relevant for forest ecosystem and management. And the first one is for the carbon cycle, specifically carbon sequestration and storage. So this is an area where it's still very much a black box. Not much is known in terms of really in different environments, and definitely not at a global scale, what's going on. So what proportion of the soil organic matter is made up of this microbial derived necromass, and what exactly what the store capacities or the current storage rates are, how it's affected by climate change in different environments and so forth. But it's a very important area of research that's that needs to be done. Obviously, now with climate change and other issues that, the hot topics of the day, basically, and that's what I'm focusing on here mostly. It's a few main points and just scratching the surface. But basically ectomycorrhizal fungi, again, focusing on this group, they definitely do contribute significantly to the soil organic matter pool, just from turnover. I mean, the incredible amount of biomass in the soil, these things. They can also, some of them anyway, are also accessing. So they're actually getting nutrients from this pool, getting nitrogen and phosphorus mostly, and in the process breaking down these more complex molecules, leaving the carbon behind and other aggregates that then is more difficult for other microbes to decompose. So in various ways, they play a role in a major role, I'd say, in carbon storage and sequestration, not only through sheer biomass alone, but also in terms of putting long-term storage. So they extend the lifespan of roots, for one. They have chitinous cell walls, which is more resilient than, say, cellulose. Both are quite robust and require microbes to break them down, but chitin even more so than cellulose. And through other biophysiological pathways, play an outsized role, I would say. Again, you'll get wide-ranging estimates, and you have to be brave to even estimate. But what we do know is it's an incredible amount of biomass, to the tune that even a conservative estimate, from example, the UN, puts it at somewhere like 600, more than 600 billion tons of carbon in the top 30 centimeters of soil. That's more than double what the atmosphere is capable of storing, and about 100 billion tons more than all of the vegetation on Earth combined. And this is, again, a conservative estimate. I expect that as our models and information gets more, as we start to fill in these knowledge gaps, that the role of fungi in general, not only mycorrhizal fungi, but also saprotrophs, the ones that are breaking down the litter, will become more appreciated over time. Another aspect that I want to touch on is the concept of forest stability and resilience. And this should also not be new to any of you, those studying forestry anyway. This figure should also not be new. This is from C.S. Halling, 1973, who proposed this concept. And basically, the circles, these little balls, are an ecosystem. The breadth of this pit where the circle is basically the thresholds of that ecosystem. So you have certain forces, maybe stressors, human manipulation, whatever, that's sometimes pushing ecosystems towards the brink of that threshold. The ability to withstand, so the buffer capacity is the resistance, so the ecosystem resistance. The height is the resilience. So the height of these peaks is the resilience. On one hand, it's the ability of the ecosystem to withstand, so to buffer away from the perturbation or disturbance. And on the other hand, to respond to return to that state of equilibrium. And so instability, it's not the same as resistance either. So stability, the ecosystem wants to return to the same state, the same ecosystem, same equilibrium. Whereas resilience, that's the system's ability to cope. So not necessarily return to the same state, but it maintains critical functions. It might not be the same species, for example, but still the same emergent properties come out of the ecosystem. And it does through self-organization, through rebounding after disturbance, and through resistance and other factors. So I mentioned the biodiversity. This is one example. These are pictures I took with a stereoscope of ectomycorrhizal root tips growing on oak trees in a tree nursery. Tree nursery somewhere in Brandenburg, we'll just say that, where the trees are transplanted every 5 years. So highly disturbed system. You might expect it to, over time, cause negative impacts on the community and things, but just this one tree species still has lots of diversity of ectomycorrhizal fungi associated with it. And if you could really zoom in, you'll see some of these are really cool. They're shiny, they can be like metallic, silver, some are pink, some are white. Just, yeah, everything imaginable, like a coral reef. You know, some are hairy, some are smooth. Lots of different types. But why is biodiversity important for ecosystems? Well, we know that, for one, it creates heterogeneity, and which in itself has its benefits, but it also provides different services, support services, regulatory services. And mycorrhizae provide such services, even cultural services. If you think mushrooms, yeah, I mean, the cultural service, a non-timber forest product. Also for the other animals and organisms that feed on them besides humans. Support services because they provide nutrients and water. They help plants establish and grow and improve their resistance to stress and defense against pathogens or root predators and so forth. And regulatory services, we talked about the carbon cycle, but there's another aspect that I want to touch on, and that is through interaction networks. So interaction networks can be multipartite with different organisms and things like that, but there's also interaction networks occurring just between the plants and the fungi. And we refer to these, you might be familiar with a mycorrhizal network, this term. A mycorrhizal network is simply when two roots or when two plants of the same or different species are connected at the root through these fungi. So one fungal individual growing on the roots of two different plants, and a network forms. And all the benefits that I've mentioned about mycorrhizae individually can then be extended to these mycorrhizal networks. So now you not only have the fungi helping a plant get water and nutrients, but they're actually transporting these between plants. This was first shown in the field in the late 90s. I think it's on the next slide here by Suzanne Simard and other persons in a field experiment where they used dual labeling of radioisotopes and looked at the transfer between paper birch and Douglas fir trees. And they had cedar as a control because cedar doesn't have ectomycorrhizal fungi. And they found that these isotopes were predominantly transferred through a mycorrhizal fungal pathway. They didn't actually look at the fungal pathway, but it was through the control species assumed. So some are going through other pathways, through the soil, through release into the soil and reabsorption, and so forth, or root grafting, for example, for trees that graft their roots. But there's now loads of evidence that it's also possible to transfer ecologically relevant amounts of carbon, nitrogen, water, and other micronutrients and so forth. I also wanted to clarify, you might hear me refer in this talk to fungal genotypes, fungal genets, mycelium. This is all referring to basically a distinct genetic individual. So it's assumed to be spatially continuous, a genet, in comparison to like fractioned ramets if something's disturbed by an animal that digs up the soil, or grazing nematodes. This could potentially break this network pathway. But when I referred to in this talk, when I refer to genets, we're assuming that it's a spatially continuous and contiguous link between the plants. So going back to the topic of ecosystem resilience, maybe now you can start to see also that it's not just about the direct support services that these fungi provide that can increase the stability of a system, but also the regulatory services, the ability to redistribute nutrients from a rich patch to a poor patch. There's different camps in the mycorrhizal science groups, ranging from those that prefer to see it like a capitalist system, where it's tit for tat, and the other camp, the more socialist system, where it's just following concentration gradients and everything's sort of transferring according to the concentration gradients. Actually, there's evidence for both. And when you look at the diversity, of course, you're going to have both depending on the species and the context and so forth. There's mechanisms, both plants and fungi have mechanisms to control the symbiosis, but also the transfer of molecules across the interface. And on the other hand, there's evidence showing that resources can transfer along a concentration gradient using like shading experiments, for example. There's a study in the east of the US that showed that trees in the overstory, or let me say it this way, plants in the understory were supplying net carbon. So there was a net transfer of carbon to the trees in the overstory. This is a deciduous forest in early spring before the leaf emergence. And then for the rest of the growing season, the net transfer was going the other direction. Similar studies have shown using radioisotope labeling net transfer based on shading, based on nutrient, and the richness of the nutrient patches and so forth. So what other implications might you think of? Let's think of Germany and forest management in Germany, for example. Anyone shout out you're designing management strategies? Considering what I've said now about mycorrhizal fungi, the symbiosis, and these mycorrhizal networks, what are some things you might want to consider in forest management? More research for sure, yeah. And I'm not looking for any big, hard, complex answer. It can like really simple, yeah. Right. So to disturb as little as possible, either the fungi themselves or the networks and so forth. Right. So this is a good example. Continuous cover management, like you have typically in Germany, is an example where it would temporarily, so over time, conserve these communities, or you maintain the current state of the ecosystem anyway. Yes. The retention is that, so I think so an old growth tree to retain old trees as so future inoculation sources for then regeneration. And so this is also true. And a very good point when it comes to mycorrhizal networks, especially, which are expected to have a big influence on natural regeneration capacity.
There's also something online, and I encourage you to wait for me to give you the microphone so everyone online can hear what you're saying. That would be great. So Alexander is writing, planting tree species that share the same mycorrhizal fungi species. Yeah. Comment.
So yeah, so either you could look at it both ways, actually. You could say, well, I want to plant mixed tree species to increase the diversity, because you'll have some generalists and some specialists within there. Or you could say, I want to plant all the same species to ensure that you're getting these networks between the specialist fungi. You could go either way. I would say if you're planting a single tree species, then it's important to think about heterogeneity over time. So for me, I think an important factor is simply heterogeneity. So rich structure forests, either spatially or temporally. So in this case, you'd have mixed age species or mixed species forests. Sorry, I think there was a hand up over here somewhere. Yes.
Mix is better for mycorrhiza as well, and the trees because of the nutrients, isn't it? So a mixed stand, because if you just have a monoculture and they all share the same mycorrhizae, sooner or later there will be maybe less nutrients for the trees and the mycorrhizae and bad soil.
Yeah, so you're getting now into another aspect of the heterogeneity, and that's utilization of the different niches which are available. I think this is what you're saying, correct? So yeah, by promoting this diversity, then you're also able to access a greater variety of different microhabitats and so forth. Yeah, all great points. Another?
What about the reforestation of devastated areas? So where you have just bare soil, can mycorrhiza help, for example, reforestation?
Yes, and in fact, some of the early field work done on mycorrhizal networks specifically was looking at establishment in in Japan on after volcano disturbance, so early establishment where there's no propagules already in the soil. So that's a bit more of a complicated question. So it was about, sorry, should I repeat the question or was it heard?
No, there's another one online, but if you want to go on first.
You know, in Scandinavia, for example, they tend to have larger clear-cut areas. And trees go back there, and there's a mycorrhizal community on these trees. So it's not like the new trees they plant aren't going to encounter spores or other inoculation sources. But you're basically hitting a reset button. Anytime you completely clear forest, whether it's clear cut or whether it's a devastating wildfire or a wind throw event, whatever, you're hitting a reset button that puts the ecosystem out of that equilibrium. And it might, what you get growing back, it might not be the same ecosystem. It might not even be the same, have the same properties. You might have something very different, a completely different state. So if you think about climate change and some of the risks and stuff, maybe it's a good idea to leave some of these legacy features in the soil to ensure continuity over time, to ensure that it grows back into an ecosystem with the properties that you want. I think maybe we move on now, or?
Okay, but there's one online also, so
okay,
I wouldn't skip them.
We don't want to ignore the online participant.
So Carlo is writing, Douglas fir is known for its relative drought resistance, even in comparison with pine trees. Also, it has been observed that Douglas fir also struggles to grow in drier conditions and artificial plantations. Could one factor be the lack of beneficial mycorrhizal networks?
Complicated question. So if I understood correctly, it struggles to grow in some drought conditions, or when it's planted as a plantation?
Yes. So also it has been overserved that Douglas fir also struggles to grow in drier condition in artificial plantations. I think it's kind of monoculture. Yeah, overserved, like overused, maybe is what Carlo means too. Observed. Carlo means observed.
Okay. Yeah, so Douglas fir is a relatively resistant tree to soil moisture stress. But it also depends on which Douglas fir we're talking about. So in North America, for example, the type of Douglas fir that they plant now in Germany is coming from North America. And you have a very distinct, of course, they hybridize, so it's not completely distinct, but yeah, you have different subspecies. You have an interior variety, which I actually studied in the past and we'll talk more about later. That's more drought resilient, and you have what there grows in more, even in temperate rainforest conditions, in mixed tree species environments, grows bigger, faster and stuff, but might not have the same drought tolerance. So, but to answer the question, yeah, we don't know to what extent that would be due to the mycorrhizal networks. Pretty much when a tree is growing in nature, you can just assume that it has a rich mycorrhizal fungal community. The things we do might affect the composition of that community and the richness, but pretty much if a tree is growing, it's going to have mycorrhizal fungi and it's going to have more than one species on it, unless it's in a test tube. Okay. So, yeah, I would just want to give one example from Europe. This was a giant study. It was published 5 years ago at the continental scale, where they wanted to get at some of the mechanisms behind the occurrence and abundance of these different fungal. So the richness. So what's driving the species richness across Europe? And they looked at 38 biogeoclimatic variables based on ICP forest level 2 data. So if you know the ICP forests, Thunen Institute here is also involved. They have long-term monitoring plots established across Europe. At level 1, they maybe measure some basic vegetation characteristics. At level 2, they're starting to get into also biogeoclimatic variables. So yeah, nutrient deposition and climate and geography, as well as the biological aspects like plant host. And giant study, giant amounts of data, a lot of statistics and modeling. And I just want to highlight some of the key findings. First, they confirmed that yes, environment and plant host explains most of the variation in this mycorrhizal fungal diversity in these forests, which isn't surprising because we know that they're dependent on their host plant. Again, about 50% generalist, 50% specialist, and some like 10% even facultative mutualist. But this relationship with the host is going to mean that you're not going to go out and find porcini growing in an acacia forest. You know, mushroom hunters know this for a long time. They know which tree species they have to, in which forest types they have to search for the fungi. And this isn't because the mushrooms only grow in this certain microenvironment. It's because the ectomycorrhizae that are producing the mycorrhizae, producing the mushrooms, grow with a specific host. So they attribute it to these main factors, but overall, you know, it's still, the model is only explaining, I think, if I remember correctly, about 40% of the overall variation. So there's still a lot of uncertainty, of course, in these mechanisms. And one factor that they left out is time. So we know that the mycorrhizal community changes as the forest develops, so different developmental stages. Not only that, but through the seasons. So you tend to have periods. It basically follows the mycorrhizal fungi. It tends to follow the productivity of the trees that they grow with. So when trees are younger and they're growing really fast and very productive, then you have a greater amount of biomass of these ectomycorrhizal fungi, so greater root tip abundance and maybe even more turnover, so higher turnover, compared to an old growth forest. Or, different in a season. So in July and August, you tend to have more biomass of these ectomycorrhizal fungi living and exploring the soil compared to the middle of winter or during the driest parts of the summer or early spring, for example. So this can also affect your results of when you take the sample or in which developmental stage you take the sample and so forth. It's not just a matter of, you know, taking it from an oak tree or a pine tree and comparing the results. So do we need a break? I didn't actually check what time we started because of the delayed start, but if we need a break, we can take one now. Otherwise, I'll go quickly through the next section and then we can take a break. Break time? Okay. So I want to talk a little bit now about experimental design. And again, I won't go into depth, but I just want to cover what I think are a few important points to consider, which are specific to the mycorrhizal systems and maybe a bit different than plant or animal experimentation systems, which you might be more familiar with. So in the first box, you see some basic measures. And I just want to say that it seems basic. Identity, abundance, you know, these are like basic measures of an organism that you want to study. But with ectomycorrhizae, it's still a black box. I mean, it's literally a black box. It's dark down there. You don't see anything. You need to take it out and look at it in the laboratory or to extract the DNA and to analyze it. And then you get the information later. So some of these basic measures like identification and abundance and things are still, huge knowledge gaps in this research. So then I'll talk about, issues of various dimensions of scale, experimental design, and then models, but just briefly, so don't worry. For identifying fungi, again, there's kind of two camps. You have the ones that look at the morphological or anatomical characteristics, and then you have the ones that maybe look at molecular based identification, and then many use both. Of course, both produces the highest resolution. On the left, you see here a morphological based approach where this is all one species. So different, you know, they're slicing up a root, staining it with different stains. This, I know you can't read it, but this is just a list of different stains and reactions that you might expect, and all part of a classification system for just the root tip part of the mycorrhizal fungi. So not even getting out into the mycelium and the extra-radical hyphae and so forth. And you can see this is all for just to describe a tomentella species. So quite big detail. You can also do rough morphotyping where you're just looking at, okay, color, does it have hair? Is it smooth? Is it rough? And things like that. Get it into a broad category and then extract the DNA and identify it with sequencing or another approach. And of course, on the right is an example of the PCR steps for a basic Sanger sequencing method. And with the advent of PCR in when was it, the late 80s, early 90s? Anyway, in the mid 90s, you could say they began developing useful markers for fungi, ones that are variable to a degree, sort of at the species level, they're variable, but then conserved at higher taxonomic levels. And this really opened up this black box for community studies of ectomycorrhizae. What used to take ages, I mean, imagine going through all these steps just to look at one root tip. And in one soil core, you're going to have, you know, hundreds of root tips. So you can spend an entire day behind the microscope just to describe one root tip. And I don't know about you, but after 8 hours behind a stereo microscope, I start to have panic attacks. So the molecular revolution, as they call it, really opened up this black box and allowed, you know, just like it did with plants and some other organisms, it's really, everything's being reorganized, reclassified. The number of species has exploded because what used to be thought to be all one cosmopolitan species has been shown to be actually cryptic species. So individual species which just look alike. And let's see, what else? Yeah, so different measures. Typically you go for ectomycorrhizal systems, you might go to a forest. If you're doing field studies, you take a core of standard size, take this core full of soil back to the lab, you wash it out, you wash gently then the roots, maybe cut them up into 1 or 2 centimeter lengths, and then you begin to pick out these root segments and look at them under the microscope. Or in this case, in the figure, they use a line intersect method where they're actually counting the tips that maybe intersect with this grid. I think nowadays it's probably more common to slice them up and take a certain amount, say 100 of these root segments. And hopefully if you take 100, then you start to reach that plateau of the species to sampling ratio. So the more number of samples, then you eventually stop encountering new species, or should, if you want to have a good saturated sampling of that system. Of course, now with high throughput sequencing, sometimes you can even just skip these steps and just, you know, put it straight from the environment. You can extract the DNA in bulk and then use specific markers to go through and let the machine sort out what the different species are by matching it to known sequences. And also full genome sequencing has really become within reach in the last decades. So that said, think about if you want to do a field study, there's a difference between, of course, sampling and a pure census or a observational sample versus experimentation. In experimentation, you want to test some treatment. You have a predictor. You have a variable that you want to predict, and you want to know what effects does this treatment have on that variable. So you want to get rid of all the other uncertainty except for what you're manipulating by with this treatment or a group of treatments factor, let's say. So you're basically getting at cause and effect. The point there is to get at mechanisms, and you want to try to reduce the other noise. Whereas sampling, you're just, you know, you want to see what's there. So of course it's relevant to think about in mycorrhizal fungi, what is the sampling unit? You know, if you want to do an experiment, what is the unit that you apply the treatment to? In the background image here I just took from Google map showing various, it's a patchwork of logging operations in British Columbia, Canada. So maybe you want to study, for example, what the effect of different management applications is on the mycorrhizal fungal community. If so, what is my sample? Is my sample the trees in the stand? Or is it the root segments that I'm collecting from the core beneath the tree, or something else? You can answer if you want. It's something else, it's the plot, yeah? You're applying the treatment to the plot, not the trees in the plot. And this is important because if you think about it, one core, like I said, you maybe pick out 100 root segments from this one core, but to get a representative sample of the plot, you're going to have to take multiple cores. Because one tree might have a completely different community than the tree next to it because of the spatial heterogeneity and temporal and so forth. So you want to take a representative sample of cores. you want to have a representative sample of roots within those cores. But at the end, your sample is only the plot. So you have to have replication at the plot level. So very quickly, you're getting into thousands and thousands of samples, or you're looking at thousands and thousands of root tips, you could say. So it's important to consider that when designing experiments. Of course, you also have the aspect of how natural the conditions are in these experiments. So in vivo is simply you're observing something in its natural habitat. In situ, then you want to manipulate, introduce some manipulation, but it's still, you want to see how it reacts in its natural habitat. And of course, in vitro, as you know, is then really limited in the laboratory microcosm, test tube, for example, where you're really excluding all the other variables. Might not have an immediate ecological relevance in a greater context, but you're maybe getting a very specific cellular level, basic biological information or physiological processes and things like that, other mechanisms. So in that respect, in the mycorrhizal symbiosis, there's various aspects to this, sampling and grain and extent. Sorry, if you've taken your GIS courses already, this should also not be a new concept to you. But it's a bit different because now you're talking about, just like with other animal systems, for example, you have the taxonomic levels, trophic, spatial, temporal. Then you have genotypic, morphotypic, functional traits. Functional traits you might get at with, say, stable isotope analysis. Genotype you'd be getting at with, depending on the scale again, whether it's you're looking at individuals within populations or communities or meta-communities. There's different molecular methods and different approaches you take depending on what your target is, obviously. And right now, hot topics, I'd say, are still continue to be genomics, metabolomics, so these big community kind of studies. And for this reason, bioinformatics is a huge skill if you want to study mycorrhizal fungi. And there's a lot of variability and stuff, but there is one pattern that seems to emerge in temperate ectomycorrhizal forest systems at the community level. No matter where you sample, no matter what tree species, what environment, there's a certain pattern that emerges, and maybe you recognize it already from these figures that I put on the slide. You see what it is? So these are all showing community level assemblages, so species richness of ectomycorrhizal fungal units. It might be species, it might be operational taxonomic units based on genomics or so forth, but you see that they all follow a similar distribution. We have a peak at the very beginning and then a long tail. Peak, long tail, peak, long tail. So obviously the axes are different on these, but basically you have abundance or frequency on the y-axis here, for example, and then the number of species shown, called the singleton, so that long tail. You have a few species which are really abundant and dominant of the sample, and then you have a lot of just, okay, you encountered it once. A lot of these are remain unidentified. You can't match them to known sequences and so forth, and they show up in every sample. You always get this long tail of stuff that you're just like, well, we'll just put that over there in this group, and we'll focus on these abundant ones that seem to be, important in the system. But importantly also, like I said, one tree might have a completely different species assemblage as the tree next to it. So if you want a representative sample of the forest, you really have to take a lot of different samples because of that spatial heterogeneity. You'll still have the same pattern coming out where you have a few dominants and lots of singletons, but the species composition differs. So my point here is that if you want to design or get into a study these mycorrhizal fungi, I would highly advise you to consult with an environmentrician or a biometrician in advance when you're designing the experiment, to already start to think about, okay, what methods am I going to need to use to analyze the data? Because some of the classical statistical approaches make assumptions about normal distributions and things like that that just don't fit ectomycorrhizal fungal communities. That's why you get a lot of like, multi-dimensional scaling and nonlinear models, mixed models. So you have autocorrelations, spatial autocorrelations, canonical correlations, where you have interactions between the predictor and response variables, for example, and so forth. So I would encourage you to, unless you're an expert in statistics and data analysis yourself, to consult early on when designing the experiments. And start thinking about, okay, what data do I want to collect? What are the measures, and how am I going to analyze it afterwards? Yes. So now I give you a break. Shall we take 10 minutes for time? So.
Yeah, nice. Then take 10 minutes, and at 5 after, like at 6:35, we meet up here. And this is also a good point to advertise here some drinks for you on donation base. So grab your beer or lemonade if you would like to drink something. And yeah, the rest goes to IFSA. Woo! Yeah, and we have some time for questions and a short discussion round.
Hello, Anna. We were just sitting here and reading your question.
We should say, I don't know what study program you're in. We could say Christoph Nowicki is the program coordinator.
No, I think this is an international student, and we have a backlash.
Okay, welcome back to the second part of the lovely lecture for Mycorrhiza. We're diving now into the topic of mycorrhizal networks, like 15 to 20 minutes. We have to cut it down. It's difficult because it's like a really interesting topic. So, but Mr. Beiler gives his best for it. And afterwards, we have like a discussion round, and yeah, then you can ask some questions. We can dive into like specific topics you want to ask, and yeah, stay tuned for the second part.
Oh, thank you again. Is Mike's on? You can hear me? So in this next section, I wanted to talk a bit about my personal adventures with ectomycorrhizal fungal research. And I'll try to focus on this mycorrhizal network aspect, which has probably been the most controversial of my studies, or topics in general. I'm not the only one that studies this, but yeah, I'll try to keep it then concise to leave time for questions. But before I go there, just to say how I got there. I was always interested in mushrooms. I thought maybe, mycology, ornithology, or maybe mammalogy. But it was through volunteering in Arizona with the Game and Fish Department on a study with squirrels that it really captivated my attention. The mycorrhizal symbiosis. And it was this squirrel here, shown here. It's called the Ebert squirrel, or tassel-eared squirrel. It's endemic to that part of the Southwest U.S. in Arizona, is where I was studying my bachelor's in northern Arizona. And basically, they were completing a 10-year study on these squirrels. Why these squirrels? Because the squirrels are important for Mexican spotted owls and northern goshawks. So the squirrel was an indicator species for more or less sort of the health of the forest habitats there in general. And these forests, this was a ponderosa pine forest, and this is the largest contiguous forest in the lower 48 of the US. Which is, for some, surprising that the desert Southwest has the largest contiguous forest, but so it is. Here it is in northern Arizona. There's the Sky Island there, and that's where the study took place. And the squirrels are important for the trees because the squirrels are dependent on the mushrooms produced by these fungi, ectomycorrhizal fungi, truffles and false truffles. And the squirrel populations go up and down along with the production of these truffles. In turn, they consume them, they spread the spores. These spores then provide inoculant for future trees, or the trees there. And the food source for the squirrel, so then the squirrel disperses the spore. The ectomycorrhizal fungi depend on the trees to grow because they're mutualists, and the trees, of course, also benefit from the mycorrhizal fungi. So it's this three-way symbiosis. And they were studying it because at the time there was a number of catastrophic wildfires, and the management regime was to thin and burn the forest to restore it to more historical conditions, so more open park-like canopies. And they were studying what the effects on the squirrels were. And I asked the question, well, what do we know about the effects that it's having on the ectomycorrhizal fungi? Because it was showing that squirrels are dependent on them and so forth, and there was no answer. So I took it up as a study project. And that's how I came in to study mycorrhizal fungi. I won't go into the details of that study so much. I'll jump right into, then I continued in higher education. I went to Western Canada, and through my previous study, I'd learned about the phenomena of mycorrhizal networks. It was also intriguing, and it was a good forestry school, so I decided to go to Vancouver, Canada, and research there. And while most of the research that had been done to that point had focused on resource transfer without actually looking at the pathway. So the pathway was assumed through experimental controls, but no one had actually looked to see if the fungus is connecting the plants. Right around that time, the first study to do this came out in Japan with Matsutake, the one I referred to earlier, and they found that they started to map out these fungal genotypes, as well as mapping the trees where the roots came from. So they were using, I'll get into this more in a moment, but a genetic barcoding method to identify both the fungi and the trees from these mycorrhizal root tips. And I used the same method then later. So anyway, this is a map of British Columbia showing the average moisture, the rain per year over the last 50 years or so. And it's just to say that this is a dry, cool Douglas fir forest zone. This is interior Douglas fir again. So Sudatsuga menzizii, the variety Glauke. And they look like this shown in the picture. So the typical is grass understory, drier environments. They grow slower than on the coast in monotypic Douglas fir stands, but they usually have these mixed age forest types as shown here. You can see because of the seed events, you don't get a continuous regeneration. You get pulses of regeneration, and this results in so different cohorts of trees, multi-cohort stands, and you see this in the diameter distribution in the histogram. We'll have multiple peaks, and you can use that then to in most cases, identify the cohorts. I studied, I chose two closely related species of ectomycorrhizal fungi. They look exactly the same. And I chose them for a number of reasons. One, they had been largely studied. There were molecular markers available to identify them. They had recently been discovered to be two separate species, whereas before they were thought to be a single species. And they only grow on Douglas fir forests. So it gives a nice, simple system because you have obligate mutualism. So you know that if you sample the Douglas fir roots, these are abundant across all the Douglas fir forests that have been sampled to that point. So you know that you have a high likelihood of encountering them. You can recognize the root tips in the field because they're big, not like a lot of them where you have to wait and look in the microscope. And they only associate with Douglas fir. So a nice candidate for making these networks. Other characteristics, as you can see from the root tip, they form these massive brain-like interwoven structures where the root tips, they swell up, they interweave, and then this whole thing is encased in a fungal sheath. So again, if you think about networks, things like energy networks, and it already seems like this fungus has some like networking properties. This fungal sheath, it's like the energy center where the energy is exchanged between the plant and the fungus, and it has this big ball, a lot of interface that's completely encased in the fungal sheath to protect it from outside stressors. They also form highly specialized rhizomorphs, which I talked about earlier, that are very good at transporting water and nutrients. And these are dry sites. So again, ecological significance. And for all these reasons, I chose to study these two sister species. This is a picture of what it looks like. I just pulled a rock out of the ground, and you can see a mycorrhizal root tip was plastered to the side of the rock right there. And this is a very typical place where you would find these species. On the other side, if you can see it, maybe you see some brown and white emanating hyphae and rhizomorphs there along with the roots. Actually, this is an immature spore carp. They form false truffles. But it also shows the rhizomorphs attached to it as an example. And I mentioned the genetic barcoding method. This was the same thing used at the time in forensic analysis, and I think it might still be used in these genealogical tests you can mail in. It's microsatellite DNA analysis. You might know of it from plant sciences as simple sequence repeats, but it's basically like repeated segments of DNA. They don't code for genes, but they are highly variable to the point where you can use them to distinguish individuals in the population. And this is why they use it in forensics. And the idea was, so this is, you use a variety of markers, and this is what's called a multiplex, so I won't get into technicalities, but basically each of these colors represents a different marker, and you have a certain amount of these markers that gives you a confidence that you're getting an individual. So here in the top you see 3 tree markers, and here again the same colors. So here you have green, you didn't at the top, but this is a different tree, so 2 tree genotypes. Whereas the other marker shown here without the stutter banding, as they call it, is the fungal markers. And if you look at those, they all match up. So this is an example of one fungal genotype that was associated with 2 different tree roots. Because again, from the mycorrhizal root tip, you're getting both the tree and the fungal DNA. And then from the tree DNA, you can match it to the trees above ground, which you then get from cambium or needle tissue and so forth, the genetic material. So that was basically the crux of the methodology that I used to identify all the individuals which I'll be talking about. I show here fishing nets. Why? Because we talked about the sampling grain and extent. Well, imagine going fishing but not knowing what you're fishing for. Use too big of a net, you're going to have little fish slipping through. If you use too small of a net, maybe you don't catch anything. Maybe you don't like the fishing analogy, but I thought it was good because I didn't know what I would be looking for, and therefore I used a variety of different, I targeted different spatial scales, different scales to try to make sure that I was getting a full picture of what's going on in the soil. And I also list the objectives right away. So first of all, I wanted to know individuals. What are the autoecological traits? So what habitats are they living in? Why do these two sister species coexist even? Like, how do they coexist in all these sites without just outcompeting each other? And what are they doing? Are they forming continuous links between trees at a very fine spatial scale and so forth? So fine spatial scales, a little net. Then I wanted to look at the forest stand scale, who's connected to who. So what's the topology of these potential mycorrhizal networks? And then finally, looking across the landscape in repeated plots, I tested 2 different soil moisture regimes as a proxy for increasing site water stress with climate change, which is expected in that region. Soil moisture regimes here, you can use slope position as a proxy. So they have very characteristic plant understory communities. At the upper slope position, it tends to be drier. You have more pine grass dominant system. And at the lower slope positions, they're more moist, and you have more forbs and feather moss and things like that. So these were the indicators that I used. I had sites that were separated by at least 150 meters, but all on the same mountain. So not so far apart that you maybe get other factors influencing the variability. And I started off like a good archaeologist and started excavating the soil. I decided on a sampling approach that looks like this. So it was contiguous soil blocks that were excavated in 2 cardinal directions, 2 meter by 2 meter, I believe it was. And then, also did the soil layers separately because I was copying a sampling technique that had been used by a group in China already. And I met the guy at a conference and asked him how he managed to get these nice soil blocks sampled and stuff, because we always use cores. They always break when they hit stones. They're difficult anyway. And he used one of these chisels, so a hammer and chisel. And I thought, with soil? Wouldn't work. It works really well, actually. Talking about this tool here. Anyway, I told you I'd be concise. I'm trying. So I decided on this sampling approach for the fine scale. And I basically wanted to know, okay, are things contiguous across space? How prolific are they? And what's the difference between the 2 species? And then, of course, I contrasted those between 3 sites having moist soil and 3 with more drier soils. And they were basically found at all the sites. So they were more prolific in the moist sites compared to the dry, but they were everywhere, they were contiguous, and they connected a lot of trees even within a small space. So already within 20 centimeter by 20 centimeter space, I was finding multiple tree roots attached by a single fungal individual, which is good evidence that these connections actually exist, that they're not, you know, just clones that are not, that are actually separated spatially, you know, that you find a clone on this tree and a clone on this tree. So this is showing that the connections actually exist in close proximity. So here I say 13% of all links within 20 centimeters, and 33% of all the links between the trees were found within 40 centimeters. So again, close proximity. And I won't go too into detail about the numbers, but it was a lot of trees for such a small space. Probably the most interesting thing that came out from this study was that I found the two species occupied significantly different depths in the soil profile. But I had done other habitat affiliation metrics and stuff like that and found that actually they both prefer the same habitats. So how does that work? Basically what's happening is, and you know, it's not like I could just tell this in the field. Like I said, they look the same. So it's only, you know, a year later when the lab work is all in and the data's there that this picture really emerged. That when they were occupying the same space, one was going deeper in the soil profile than the other. And this, on average, drove that significant difference in depth that we observed, as shown here. So what this looks like, this is just a soil pit from one of the sites, and you see they both actually preferred the interface between the forest floor layers, or organic horizons referred to, and the mineral soil horizons. So this interface, they both like to hang out here. But when they occupy the same space, one goes into the mineral soil and the other stays above. A group in California went on to test some different hypotheses about why, so the mechanisms behind this, and they found that sure, there's some competitive interactions that go on, but also about who gets there first. So like the one that tends to stay at the top, if it gets there first, it has an advantage. But otherwise, the other one, yeah, tends to go around, and I'll get more into that in a moment. I also sampled a whole area that was like 1 by 2 meters just to get a more higher resolution picture of this, and that's shown here. So you can see the Vesiculosis, shown here, was more prolific across space, larger genets, but it also grew deeper. So the red here is deeper, the purple is absent, and the blue is where it was more shallow in soil profile. And because in this particular one, it was sampled to a 30 centimeter depth, so I did it in 3 layers. So 10 centimeters at the top, that interface then basically, and then the mineral there below. Another picture of how it looks there, the distributions. So moving on to the forest stand scale. Then I wanted to know who's actually connected to who, speaking about the trees and the fungi, of course. And why? It's because different network topologies, and this was quite a new emerging thing at the time because of the internet and different real-world networks that they were discovering that they all shared similar qualities. And that's shown all the way on the right here, what they called a scale-free network. Biodiversity and Albert, I believe, coined that term. But basically, some nodes in the network are much more highly connected than average. And this makes a node degree distribution that follows a power law curve. So a power law to describe the distribution of links among nodes in the network. The structure of the network can also give other insights, like how robust it is to attack. And I'll get more into that later. But basically, I just wanted to know what it looks like. You know, is it nearest neighbors are connected, like a regular? Is it random, where nearest neighbors are connected, but then also easily traversed from one side of the plot to the next? Is there no connectivity? Could have also been an outcome. Or is it more complex? That's just also a crude stand visualization system. I don't think any of you took the digital twin forest specialization module with me last semester, but you would laugh if you did because of this crude stand visualization. But anyway, I wanted to show the distribution of the size classes in the plot. And I began toying with different ways of visualizing that. What I decided on was just a basic GIS top-down view, like shown here. And maybe this graphic is what brought some of you here. This is the one that's been reproduced quite a lot. And it looks pretty, I have to admit, it's complex. But I want to step back now and walk you through the steps of what leads to that complexity. So first you have the trees. And here the circles are sized according to the tree diameter at breast height. So again, you can see that there were different sizes and ages of trees in the plot. I used a non-random sampling approach because I wanted to ensure that I got the roots from even the small trees equally likely as the roots from large trees. So what I did was I sampled the 4 cardinal directions within the drip line or around the drip line of each tree, in addition to some plot interspaces to get a good coverage of the entire, and this is a 30 meter by 30 meter plot. I don't think I mentioned that. So 30 meter by 30 meter in one of the moist areas. So then later when the genetic results came in, I could say which fungi were grouped with, what genotypes grouped together. And that I just circle in the diagram. And I could also tell, like I mentioned, from the same mycorrhizal root tip location. So these are the little dots are the sample locations, the actual location where I found the ectomycorrhizal root tip. And it's just a straight line linking it to the tree where that root came from based on the genotyping. And carried that on and began to build that network. So this shows the distribution of the vesiculosus genets. Vinicolor genets you can already see right away. Same amount of genets, but the vinicolor were much smaller. This just shows the root lengths. So the tree bole linked to the place where the root was sampled, and put it together and you get that network picture. So as you saw already, the same fungal species that forms larger genets, and we found earlier that it occurs on average deeper in the ground, also connects more trees. It links more trees, and it's a strong correlation between the size of the genet and the number of trees that it links. From a network perspective, this shows the topology. So now the lines show what trees are linked. So if there was any links between the trees, it's shown as the line. The circles are the tree, again, sized according to the diameter breast height and colored according to the cohort class. And the thickness of the line here is not just the number of links by multiple samples, but by different fungal genotypes. So if multiple samples were found to link 2 trees together, that was reduced to just 1 link. That would be a loop in network terms, and it was reduced. So here the thickness shows that these trees were actually linked by more than 1 fungal genotype. So you have this, these various levels of nestedness and complexity in the network and stuff, which I could spend a lot more time talking about, but the take-home message is that it was very highly connected. Why is that important? Well, for one, it demonstrated that these networks actually exist in nature. I only looked at 2 closely related species among hundreds that occur in these forests. So it's much more complex than what I'm showing you right now. Another aspect is the plot edge effects. So 30 meter by 30 meter plots, but we're talking about old growth trees here, where the root systems are not contained by that 30 meter by 30 meter plot. In fact, the tree that was linked to the most other trees, shown by the arrow here, was located outside of the plot boundaries. So its roots were encountered in the plot, but the tree bole was actually outside the plot. If you scale that up and think about, okay, then if I would have gotten the whole root system, then you're talking about, you know, connections to hundreds of other neighboring trees. From the fungal perspective, it means that they gain access to a long-term energy source. So again, stability over space and time, ecosystem resilience, and these concepts, it's not just about the structure, but I will mention that, but it's also just about the fact that by colonizing multiple hosts, it's essentially securing itself a long-term energy source. When an old tree dies, it still has the young ones to replace it. And on the other hand, it creates an environment for the young regenerating seedlings where as soon as they germinate, they gain access to this expansive network and can sometimes persist small there, tapping into this network until the gap in the canopy allows them to then grow into the next dominant tree in the stand. So this is the tree shown here that was connected, the most connected, I mentioned. And this is diagrams getting into more the topological structure and this, what was called a scale-free property. You can also refer to them just as hierarchical, nested, complex interaction networks, whatever you want. It's scale-free to a point, because I'm not trying to say that that this tree would be directly linked to a tree in Central Africa. But at this plot scale that we looked, this network was very highly interconnected and easily traversed. And I mentioned that this can also indicate its robustness to attack. Here you see an example when the largest hub tree was removed. You can see that, yes, there's reduction in the links, the density of links, but everything is still connected. Likewise, if I take away the largest fungal genet, there's a big reduction of links, but everything's still connected. If on the other hand, and this is all hypothetical removals of elements, but if I take out 20% of the trees at random, there's a big difference in that effect then versus if I take still 20%, but all the oldest cohort out of the picture. And this is relevant for the forestry management practices there, where traditionally they did what's called high grading. So they go in, they take out the strong, the biggest and the straightest trees for lumber, and they leave everything else. So it does have forest management implications based on the network topology. And yeah, I won't go deeper into detail because I want to leave time for questions. We're already gone over time, I apologize. This was then extended into the moisture comparisons, as I mentioned. And so you get more pictures of networks. And long story short, they were highly interconnected every plot. So I actually expected that in drier sites, it might be more connectivity than the moist ones based on the stress gradient hypothesis, which would suggest that trees would invest more in the symbiosis in more stressful environments. Maybe I didn't look at extreme enough differences. Maybe there's other reasons, but they were, yeah, highly interconnected at every plot with a similar structure. And interesting also, there's the fungal perspective, which nobody really talks about, of the network too. So you can flip it around where the fungi are now the nodes and the trees are the links, and you get a similar structure. Even those spatial distributions are completely different. When you're only looking at two species of fungi anyway, you know, you'd expect less overlap compared to the roots. So yeah, some of the quick, very quick overview. That. Sum it up, gist of the story was based on the size. So if you ask me the mechanism, what's driving all this complexity, is actually very simple. It was just the size of the organism. So the plot edge effects created some of that, you know, because it's cutting off parts of the fungi, which results in different sizes. The but you also have the comparison of the two species, where the one's bigger body sizes than the smaller one, and you get the same pattern. So the larger the body size, the larger the zone of interaction, the more connections to the other organisms, so the more larger social network it has. And this makes sense if you think, you know, a larger dog's going to have more fleas than a small dog. It's all about the zone of interface and interaction there. So both for the larger trees and for the larger fungi, are, you know, larger trees are hubs, larger fungi are hyperlinks in this example. So yeah, I think I'll just leave it there and we'll move on to questions. I see a lot of,
Yeah, I'm really sad to have to, but I have to stop you. Yeah, thank you so much for your presentation and for this second part of the lecture. I really like the graphics. We showed how everything was connected with each other, and thank you so much for the insight of this mystery regime of fungi. And now we come to the point of the discussion. And so if you have questions to our lovely speaker, we have like now some time to ask some questions. There's the light, yeah? And yeah, maybe I can take the privilege of being the moderator and ask my first question, and you have some time to brainstorm a bit. And also the people online can write questions in the chat, and Nele will gather the questions. And yeah, I'm really interested. We are all students here and studying like nature-based sustainable study programs and learning a lot of biodiversity loss, species loss. And my question is like how fungi and the species of fungi are threatened by the climate change in terms of loss of species. Like, for example, how it is like with the insects, like we have like an insect dying because of climate change. Is it also the case at fungi? And or is it now researched a lot, and what the humans can do to provide a better surrounding and to enhance the environment for fungi? We have a lot of ecosystem managers here. What can we do to provide a better environment for them?
I mean, I think, I guess we have to differentiate between, so like species richness, biodiversity versus loss of the whole, you know, all fungi. So I think fungi in general will be one of the last organisms to disappear from the earth. I mean, they're so adaptable and so much plasticity there. But mycorrhizal fungi, for example, sure, they're impacted because they depend on the tree distributions, or vice versa. The tree distributions can depend on the presence of the fungi. So it's they go together. So it's the distributions may change. They might not. Yeah, if you think, for example, with microheterotrophs, there's some orchids that if a disease comes in and wipes out all the pine trees, the fungus that the orchid depends on is very specific for that pine. I'm thinking of pink ladyslip orchid. I'm thinking of a specific example from the eastern US. And when the pine trees are lost, the fungus is lost, and then the orchids are lost. So it's you have cascading effects. On the other hand, there might be some fungi that take advantage of climate change conditions and proliferate. So it's, yeah, for one, we need more baseline data to have something to compare to, because then it's a repeated measures, you know, thing. But we can say, okay, well, we have so many species here, but we don't know what it looked like historically, and you know, then there's no comparison. Long answer to.
It's a good sign that it's adaptable, and that's a great companion for combating the climate change, and that it's not too sensible like other plant species and can adapt to the situation. Nice. So do we have more questions here?
But I know here, just one more comment there. It's a big topic right here, not only at HNI, but also at Tunen Institute and the different institutes, to the topic of what trees are the trees of the future. Yeah, the trees of the forest that are the most adaptable, that will be successful in the face of climate change. You can't ignore the fungi with that, because the fungi, they increase the niche breadth of the tree, its ability to adapt and to colonize a certain area or across the range. So it's really a critical point to consider when assessing the success likelihood of any tree species.
Yeah, I was wondering, because there was this study or like something that said in more milder climates or where the conditions are more harsh, there will be more fungi actually working because nutrients are not so readily available, right? Does this mean that in like tropical rainforests, for example, is there also a lot of fungi, or are fungi more in milder climates in the soil?
So tropical rainforests do have, they tend to have arbuscular mycorrhizal fungi. The arbuscular mycorrhizal fungi are especially relevant for phosphorus acquisition. So in phosphorus limited systems, you tend to get more arbuscular mycorrhizal fungi, and in nitrogen limited systems or water limited systems, you tend to get more ectomycorrhizal fungi. Does that answer the question?
Yeah, so in tropical rainforest, if we go into the soil, we also find a lot of, like, is the network as complex as it is here in your plots, or more complex even, or how can we compare it?
Can only speculate because the networks haven't been investigated there. So yeah, it would be speculation. But I think it's safe to assume that these networks exist in pretty much all forest ecosystems. And your original point is indeed true. So where nutrients are limited, it promotes the mycorrhizal symbiosis. And you see this if you apply too much fertilizer, it can have detrimental effects on the symbiosis, on the fungal nutrients, because the plant doesn't need them anymore. Yeah, so it can have direct effects, but it can also have indirect effects if the plant needs them less and doesn't invest in them. Yeah, so the latency, so the amount of time that the nutrients are available in the ecosystem is much shorter scale in the tropics. Things are cycling faster, turning over faster, and nutrients are being eaten up basically as fast as they're produced. No, they're still necessary because that leads to a nutrient limited system, or different strategies, you could say, different nutrient acquisition strategies.
Okay, my question is, are mushrooms or fungi also always connected to trees? Because they have symbiosis with trees, or they deconstruct trees, but I never heard of like marine fungus, like that lives in the water or something like that, but it should be possible, I guess. I don't know why they don't exist. Do they always have to be in connection with trees, or?
That's a good question and a cool answer, because actually there are fungi that grow in freshwater systems. So I don't know of any growing in saltwater systems, but fungi definitely. Mushrooms, there's mushrooms that only grow in fresh water. So like in the stream.
How do they look? Like, what is special?
They look like a mushroom. Yeah.
Like on the ground that you can find in the forest, or?
Yeah, but so a lot of mushrooms, are like saprotroph, so they're not growing in mutualism or symbiosis with the tree, but they're basically either decomposing litter or they're feeding directly on as the tree is a parasite. So like oyster mushrooms, for example.
Nice. So where do we have another question? We have some questions from the chat, but if there's someone on the way, I can start quick.
Just a little just a comment on the saltwater fungi. There is for sure mold sustaining salty conditions and being partly underwater, which I encountered myself crossing the Atlantic in a sailing boat. And every sailing boat in the tropics has a specific smell of that type of mold.
Okay, extra information from the people here. So what does the chat say?
The chat asks again, like from Anna Ackerman, which is now offline, but she wanted to have the question replied. Thank you for the info on reset landscapes. I'm currently studying mine reclamation in the southwest of the US, where this might be relevant too. How should the sourcing of, say, spores work in practice? Are the nurseries like they are for plant material?
Yeah, so there's various methods of producing inoculant. So sometimes it's with like what do they call it? You know, like bioaccumulator things. Sometimes they just use plants like leeks, for example, as to accumulate inoculant. Other times they're actually using artificial means to propagate them. But there's inoculants on the market, yes.
Okay, thank you. Then there's another question from Alexa Beaucamp. I am forest manager in Brandenburg and Mecklenburg-Vorpommern. I'm interested in the role of logging lines as we use them here, 20-meter Rückgassennetz, so the logging lines. Does this narrow net of lines thread the Mycorrhiza?
Yine. So yes and no. Obviously, if it depends. If it's in the middle of winter and or if the vehicles are only traveling on you know, piled up wood or something. So it depends partly on the mechanical damage to the soil. There can be a direct mechanical damage. If it's cutting deep enough that it maybe you know, rips roots, then it's also going to rip through the fungi. But the fungi will recover and rebuild that connection. Something else that can happen is if it leads behind deep ruts, for example, then you get water accumulation, and this can simply change the water dynamics of the site. And so they can have indirect effects. But the only direct effect would be really through mechanical damage, so tearing. Compaction can happen in city environments, for example. It's been found to reduce the diversity of, you know, the community with compaction and stuff, but the fungi, I mean, you see mushrooms growing up out of asphalt. And they're incredibly resilient creatures.
Yeah.
And these, sorry, but if you remember the picture I showed from the tree nursery, a lot of these were mushrooms that specialize in forming these network, like rhizomorphs, long distance exploration types. And they tend, and this is kind of a pattern after, if you have a lot of disturbance, these quickly build back these bridges networks.
Yeah, thanks for this detailed answer. And we have some more questions from the audience.
Thank you so much. Thank you so much, first of all, for this lovely presentation and this introduction to this world that is so undiscovered and underrepresented. So I've understood that fungi and mycorrhiza are like a massive web that connects so much and that interacts by exchanging. And so my question is, I wanted to ask you if you could go deeper into the competition between fungi, because I feel like it's kind of opposite. On one side, it's got this image of, or this, you know, I understand it as an amazing routing system and connection system, but obviously there's got other factors of, and you're also talking about competition and like different movements of fungi that they have because of this competition. So I wanted to ask you if there's like certain types, maybe between genus of trees, where you could definitely see a better networking system, or maybe there's other circumstances where there's much more competitivity between the fungi.
Yeah, I mean, it's also a good question. So one thing I didn't mention is that is this aspect of our forest management. Most of the models are based on competition, so competition between trees, and they don't account for potential other mechanisms like facilitation pathways, like these networks. That's one aspect. Our models would be better growth models if they incorporated more information about other mechanisms. The other side of that, though, is that these networks can also increase competition. It doesn't necessarily always benefit a neighboring tree to be connected to its neighbor. So there might be some instances where, for example, the presence of the networks might encourage monodominance of a single tree species in what otherwise would be a mixed species forest. There might be examples of simply where the network provides a conduit for increased competition between trees, and there's simply just not enough information to make really broad conclusions yet. But if you, like the forest where I did the network study, there's a really interesting, at the time there were also studies, sorry, I'm bouncing around here a little bit, but there's a spatial aspect involved. There's been a number of studies that show that as you progress away into from the tree bowl, there's like a zone of the highest species richness. So there's also spatial correlations like with the distance to the tree bowl itself. And right around drip line, you tend to have the highest level of diversity of these fungi. And then if you look at the natural regeneration patterns, it's very common that you see a large mature tree, and then you have a developing cohort. They're all the same age, but they're not the same size. Some are doing better than the others. And you'll often get this pattern where closer to the tree, they're stunted. And then right at the drip line, where there's less competition for light and resources, but where they're still connected to the network and have the highest species richness, that's where you get the greatest growth. And then it goes back down as you get farther away. So some of the patterns that we're getting from the community studies, where they're looking at distance from the tree and the species richness of the fungi, match what you actually see in the natural regeneration, where the greatest growth is at this, like, it seems to be like a sweet spot. So it's not just facilitation, it's not just competition, but there's a spatial aspect where at the right distance and the right conditions, it has the maximum effect of benefit, hypothetically.
May I ask another question, please?
We had, maybe let's do the 3 final questions. And yeah, there's also some in the chat. Okay, but here was also a question.
Are there any ongoing like projects or internships regarding this topic?
I would be happy to meet with anyone interested. I can give you advice, connect you to people. I don't have any current projects now myself, but I'd be happy to meet with anyone, consult about opportunities, future opportunities, current opportunities, or just ideas.
Thank you.
Okay, yeah, maybe we can share also the contact dates, how to reach out to you at the end of the presentation. And we have some questions in the chat. I would say like 3 questions more.
Okay. We can do. Okay. So Tim asks. Yes.
You said that ECM diversity is dependent on host factors. During field research in Laos, I have come across fungi of the genus Ganoderma, some of which seemingly exclusively grow on certain tree species. Oh, your laptop died. Okay.
Oh no. Sorry, was that Piloderma?
Fennel.
With a P?
Ganoderma, but I don't know if this question is maybe too specific, but maybe the other question. Okay, stop holding the camera. Based on what you explained about different fungal species being able to exist in the same space, but one being is more likely to move down in the soil, based on this interaction, I was wondering something. To what extent do the fungal species that are associated with the invasive exotic tree species compete with fungal species that are associated with native species, and therefore aid in the invasiveness of such tree species, specifically in the competition between mycorrhiza?
This could definitely, so the question was, I guess you heard it with the microphone, but yeah, this can definitely happen where the mycorrhizal fungi can facilitate a species invasion. In other cases, plants are particularly good at invading because they've developed strategies which don't depend on the mycorrhizal fungi. So they're just good invaders by themselves.
Are there some famous examples for this you have in mind, or is it this?
No, not specifically, though I've heard stories that, because Douglas fir is also an important plantation species in New Zealand, and I've heard stories that they struggled for a long time to get it established. And then at one point they introduced soil from the native habitat, which then introduced the mycorrhizal fungi, and now has become an invasive tree species. I don't know of any empirical data backing that up, but that's just the story that I heard at one point. Yeah.
Okay, interesting. Okay, Ganka.
I was wondering in which extent mycorrhiza are used by the trees to communicate with each other. So, for example, when the trees lose their leaves in autumn, are they also communicating with each other when to lose their leaves, or just exchange of nutrients? So, yeah, the extent of communication.
Yeah, so communication signals have also been shown to travel through a mycorrhizal pathway, both with arbuscular mycorrhizal systems in tomato plants, and also between, I think it was ponderosa and Douglas fir possibly, so two different tree species with ectomycorrhizae. Basically, trees are communicating all the time through hormones, mostly through the air, things like strigolactones and other volatile, chemical compounds similar to our hormone system, where we're communicating without knowing it through our, hormones. And it's communication because you have a signal, you have a response to the signal by the other individual. You can also have then, other indirect effects, like where aphids might, be on a plant. The plant sends out a stress signal, and some other insect cues in. So the predator of the aphids recognizes that signal, and comes and preys on the aphids. So there's like other examples too beyond mycorrhiza. But in terms of the pathway, yes. It can go through soil, but the mycorrhizal pathway is a more direct pathway than just diffusion through the soil. For bacteria as well. Some of these rhizomorph things, they're super highways below ground for all kinds of things.
Yeah, interesting topic also. So let's round it up with the final question out of the audience. Okay. Olicito, mach nicht zu lang.
Well, thank you so much. What a pleasure to set the last question, and I hope it's a good one. So I can't not address this topic, which I think that everybody's come across at some point. And you just briefly mentioned it. You were talking about trees, like this idea of trees being the ones that connect the fungi. And I thought that was so interesting. That blew my mind, and it made me think about this thing that this idea that fungi need the trees, but you were also talking about, well, first of all, fungi being amazingly adaptable, like the possibility to adapt to any type of circumstances, and also the proof that of the first plant exemplare, is this? Examples that we have in history, there seems to be fungi in them. And so my question is, how do you prove if this fungi is out of space and is the key component to a massive complex development of life? Because you said yourself that it could possibly be an indicator for actual plants to establish on ground or even in existence in general. What's that like? What's the point? How is the status on this question? And how could you prove that they are actual aliens? Thank you.
Yeah, good question. How would you prove that they're aliens? I think, unfortunately, we don't have the, the historical records in our, fossil to put these evolutionary pieces together. I mean, I think it's accepted in science that plants and fungi both play a predominant role in the establishment of life on Earth, of other organisms. They were the primary producers, and fungi evolved some millions of years before plants did. So yeah, I mean, on a personal level, do I sometimes picture the fungi as the sentient beings that are just simply farming the plants? Yes.
Yeah.
I mean, you know what I mean? Like sometimes I wonder if the fungi are really not just cultivating the plants like we cultivate our crops.
Yeah, that's a really interesting topic, and thanks for bringing it up. Maybe it has also space for a whole lecture, like, are fungi aliens? Where do they come from or something? But yeah, thanks for the question. Thanks for your lovely lecture. And I think it was really successful. We had like a full lecture room, and at the peak we had like 70 people online. And it underlines the demand like we have out of our curriculum, that it's not covered enough, like this interesting topic. And so thanks for filling the gaps. And I would, maybe we have a big applause for our lecturer. And if you have like further questions, I think you're a bit here, like to clarify some questions or like changing context. Maybe we can also write it down here, like the context, or send it via mail, like how to reach out to you. And yeah, thanks for joining, and have a good evening. Yeah, thank you.
Yeah, thank you. Thank you for having me.